So my name is Mohamed Al-Fageeh. I'm the manager for National Center for Biotechnology. I understand this is the last presentation, so everybody is looking forward for the subsequent discussion. So I will be as brief as possible. So first, let me allow me to express my profound gratitude to Professor Jaka and Professor Al-Maghlout, the cultural attaché, Saudi cultural attaché in Italy. And I thank ICGEB for their outstanding organization of this workshop. And I have to confess, actually, after listening to your presentations yesterday and today, and now I feel really humbled, thrilled, and quite intimidated. You guys set the standard very high for me to impress you, or at least to be satisfactory. So what I'm going to do today briefly, and I have also to confess that this presentation has been subjected to a major operation last night to remove the fatty science from it and to make it as light as possible. So what I'm going to do quickly is to introduce to you the National Center for Biotechnology in CAGST, how it came to existence, where we fit within CAGST structural organization, what we do, our duties. And towards the end of this presentation, I will tab on some of our achievements, some ongoing research projects, and hopefully by the end of this presentation, you will have at least the basic knowledge of biotechnology research to help catalyze and expedite our discussions in the subsequent presentation. So without further ado, where are we? This slide has been presented by Dr. Abdulaziz yesterday, and as he mentioned, CAGST has two main arms. This is not readable actually, so I will magnify the Life Sciences and Environment Research Institute, and the National Center for Biotechnology is an independent research center under the Life Sciences and Environment Research Institute, which also houses for other national centers. Including National Center for Agricultural Technology, National Center for Environmental Technology, National Center for Stem Cell Biology, and National Center for Genomics. So the National Center for Biotechnology is a relatively young institute which was recently inaugurated in 2010. However, research activities on biotechnology in CAGST actually dated back to 1997. When we established the cryopreservation and genetic engineering lab, which evolved quickly into an independent research program called Genetic Engineering and Biotechnology, which was the nucleus for the current center. And during the last 10 years or so, the center actually made great strides in its efforts to build a leading infrastructure for the advancement of biotechnology in Saudi Arabia. And by leveraging its multidisciplinary expertise, the center has recently begun to bear some fruit. So simply, our vision is to be pioneers in biotechnology for better life, and our mission is to excellence and creativity in innovating and developing biotechnology application, accomplish scientific and economic benefits. And we have set up some high-level and strategic goals, including to significantly contribute to the transition of Saudi Arabia into the knowledge-based economy, to establish partnership with reputed international institutions, e.g. ICGEB, and to transfer cutting-edge technologies in different disciplines to Saudi Arabia, to provide settings and to encourage creativity and investment in the field of biotechnology, to direct application of biotechnology to achieve health and food safety, security, and sustainability, to protect the diversity and natural resources of Saudi Arabia, to strengthen our relationship and ties with our community. I put this slide. This is just a brief statistics of our staff or human resources, and the main reason for putting this is just we are 52 actually, but I want to emphasize that 35% of National Center of Biotechnology staff is currently PhD students studying outside. Most of them are in UK. 10. We have 10 students in UK, 3 in US, 2 in Australia, 1 in Saudi Arabia, 1 in Japan. Unfortunately, again, I don't see Italy in this statistics, and I have already raised this point with Professor Al-Maghlout, and perhaps also we need to raise this point later this afternoon to see how we can catalyze scholarship programs into Italy, or at least into ICGEB. So we're trying to maintain the percentage at this rate between 35 to 40%, and we're expecting within 4 years we'll have approximately between 15 and 18 students coming back, PhD holders coming back and help to propel the research in biotechnology in the center. Before I proceed further into responsibilities and duties of the center, I just want to briefly mention that the CACST, in collaboration with Ministry of Economic and Planning and other stakeholders in Saudi Arabia, proposed and developed the National Center for Science and Technology. And the ultimate goal of this, by 2025, Saudi Arabia should join advanced knowledge-based economies with high competitive science, technology, and innovation ecosystem. And we have just finished the first stage, the first 5. This plan is actually 4 stages, each stage 5 years. We just finished the first stage, and it's been subjected to a rigorous review by international third party, and taken into consideration criticism and recommendation for the first stage. CACST is going to initiate the second stage, I think, within the next month or so. The reason I mention this is that during the preparation of this strategic plan, CACST, in collaboration with other stakeholders in the kingdom, have identified 15 strategic technologies, and it's not at all surprising to see biotechnology within these technologies. Not because I'm a biotechnologist, but biotechnology has been hailed as the new engine for growth, and other experts claim that biotechnology revolution will surpass that of information technology. If you agree or disagree, we have to wait and see. So the center has actively involved during to identify the strategic priorities for biotechnology, and also the center is responsible for coordinating the strategic priorities for biotechnology with different stakeholders in the kingdom. We also obviously carry out research and development activities in biotechnology in Saudi Arabia. We are considered the expertise in the scientific arm to represent Saudi Arabia in several forums, associations, organizations, like, for example, ICGEB. We provide training and consultancy for other government agencies, and we perform public outreach and educational activities. So this is just a very quick summary of our responsibilities and duties. As you know, international scientific collaboration has now become an indispensable choice if you want to progress and thrive in the field of science, especially in biotechnology and biomedical fields. And CACST strongly believe that a key ingredient for our success is to establish scientific collaboration. And during the last 5 years or so, CACST has established centers of excellence with top academic and industry firms, including MIT, Chinese Academy of Science, Boeing, IBM, UCSD, Stanford, Northwestern, Cambridge, UCLA, Oxford, UCSD, University of Michigan, and Berkeley. And the main objective of these centers of excellence is to help CACST achieve its strategic goals by providing a mechanism of developing expertise, and also by handling the current the expertise of current Saudi researchers. This program is very important for CACST, and CACST has established an independent program called JCEB, Joint Center of Excellence Program, to monitor the performance and progress of these centers. And the reason why I put this slide here, I genuinely hope that we will soon see National Center, for example, and the ICGEB. This is a wish for me. Similarly, the National Center of Biotechnology has an extensive collaborative agreement also. It's either housed by the JCEB program or an independent or initiatives, personal initiatives between researchers within the center and other colleagues, overseas colleagues. And we collaborate with the Genomics Institute. We have collaborations with the Belarusian Academy of Science. We have collaborations with the Institute of Membrane Technology in Calabria. We have collaboration with the University of Cambridge. This is under a center of excellence. We have collaboration with Northwestern University, UCSD, and UCI. And again, it is the same objectives, is to help propel and achieve our strategic goals. We consider ourselves in the national lab, so this is why we are trying to diversify our research activities to span different disciplines of biotechnology, including environmental, agriculture, medical, and industrial. And we currently have 4 active research units, including cancer biology, immunobiology, and immunotherapy, proteomics, and protein chemistry, and bioprocessing. And I'm really very proud to say that we had stem cell research unit and genomics and bioinformatics, and they have reached their critical mass and recently graduated into national centers. So now we have the National Center for Stem Cells. Although it's administratively independent, we still collaborate with them, and we have the National Center for Genomics. Okay, so now I will briefly tap on some of the achievement and research projects, and I really don't need at this time to discuss the pure science of it, but I will just give some examples. And the example is one example, the Camel Genome project. The reason why I put this as the first slide after the introduction is just to demonstrate to you how a win-win international collaboration can lead, at least in my opinion, into significant scientific impact. So this is the camel we sequence. It's a male calf camel, Arabian camel, and this guy who is involved with drawing the first blood sample from the cow. So why we are interested in camel genome? If I want to talk about from scientific perspective, camelidae or camels family is a phylogenetically divergent, representing a very interesting order Tylopoididae, which is characterized by having a three-chamber pseudo-Roman, which is widely believed that camelids evolved through convergence independent of the true Roman mammals. These animals are really fascinating because they have a characteristic physiological properties that allow them to withstand and to adapt to very harsh environments. They locate in South America, the alpaca and llama, and the camels, the two humps, the Bactrian camel and the dromedary located in Africa and Asia. Also, the camelids characterized by having an unusual immune system. In addition to the conventional antibodies, they have a heavy chain on antibodies, which has been recently exploited for immunotherapy, for example, because of their size and their stability. And if we want to talk about historical and cultural importance of camels, they both historically, camels have been an important species, particularly in the Arab Peninsula. And here I'm talking about the dromedary. And the Arabian camel is perhaps the most important species within the family of camelids, not because of their the beast of burden, but also for other benefits like meat, milk, wool, etc. And until recently, the Arabian camel largely been bypassed by the genetic revolution, although many other vertebrates have their genome sequence. So CAGS proposed an initiative to obtain a fully annotated sequence of the camel genome, including exonic, intronic, and intergenomic regions. And the objective, the overall objective is to understand the genetic component of the camel genome, including producing a fine genome map, carrying functional mutations, and performing analysis of and comparative analysis and molecular evolution. So this slide is just some envisaged potential outcomes from the camel genome, including insights into molecular basis and unique physiology that enables camels to survive and thrive in harsh environments, insight the mechanism of disease resistance, because camels are resistant, for example, to foot and mouth disease, insight and potential for discovery of new antimicrobial peptides native to the camel innate immune system, insights and into unique aspects of the camel immune system, help develop vaccines to control both livestock and human diseases, discovery of novel pathogens, etc. So we started this project in 2004, and at that time, as you know, the whole genome sequencing technology was not mature and also was very expensive, so we focused on the transcriptome, and we generated cDNA library from 11 different tissues of 9 inbred camels of 3 age groups and 3 different types of the Arabian camel. This is originated in Saudi Arabia. So approximately 23,000 express sequence tags sequence were determined from each, yielding a total of about 70,000. And we have identified and annotated 23,600 mutative genes. 29 of these genes potentially novel or fast evolving genes. We published the results in PLOS ONE in 2002, and if you are interested, you can. I will not go into much details. And then we started the second phase in 2008 in collaboration with Pigeon Genomics Institute because of their outstanding capabilities in terms of genome sequencing and informatics. And with we have been approached by the Inner Mongolia Agricultural University because they were sequencing the 2 humps, the bacterian camel genome, and we have agreed to do a full comparative genomics for the dromedary, the bacterian camel, and the alpaca genome. So what we've done is we generated high-quality genome sequence of the bacterian, the dromedary, and the alpaca using the HiSeq 2000 platform. Using combination of homologous sequence searches and from scratch predictions, we annotated about approximately the same number of genes in all three species, and the annotated genome size was comparable. We have identified approximately 12,539 gene families that share between the 3 camel-like species and the cattle. And we use the computational analysis of gene families evolution. We identified this is to study to investigate the genes that underlie the characteristic physiology of the camel that allow it to adapt to its harsh environment. We have identified 373 expanded genes and 853 contracted genes families in the dromedary, and 183 in expanded, 753 contracted in bacterian, and 500 expanded and 2,000 contracted gene families in alpaca. Then we have studied also the gene responsible for the adaptation. We have identified 287 positively selected genes in bacteria, bacterian camel, 324 genes positively selected in dromedary, and interestingly, 150 positively selected genes were common in both, which indicates that a similar selective pressure in these 2 species. We have also identified genes related to fat and energy, for example, ATP-related genes, mitochondrial lipid transport, and response to insulin stimulus. And this is not surprising given the fact that the camel live in a food-scarce environment. We have also identified genes related to stress response, DNA damage and repair, apoptosis, protein stabilization, immune system processes. genes associated with lung development, because as you know, desert has a very challenging, dust-prone environment. We have also identified possibly selected genes associated with eye development and photoreception, visual protection, with osmoregulation, osmoprotection, and water reservation. If you are interested to read more about these findings, I would kindly refer you to our recent publication in Nature Communications. So now we are building on our knowledge of genomics in camels, and we have proposed to Cox to develop a high-density SNP chip, 3 different SNP chips, a high-density genome-wide single SNP array for screening. And this project is going to be helpful for genomic association studies, etc. And we also, the second proposed SNP is a dromedary parentage SNP chip, and this is for pedigree analysis and camel identification. The other SNP is for the racing target SNP screening array, and this will be for genetic improvement of camel milk and racing productive. And we're currently actively, thank you Dr. Abdulaziz, who is helping us a lot in this. We are actively pursuing CACs now to secure funds for these 3 projects. Another research interest that we have actually in CACs is optimizing bioprocesses, and we are working towards improved mammalian expression systems. Again, I will not go into much details, but I will briefly tap on this one. This is a common knowledge slide that you need expressed protein for functional study, structural study, vaccine, for example, therapeutic drugs, industrial enzymes, etc. And this figure just to illustrate different expression systems. And in the next slide, again, this is a common knowledge, just to emphasize the pros and cons of different expression systems. We are interested in the mammalian cell expression system. It's very advantageous if you need your protein folded and correctly and post-translationally modified. But unfortunately, the disadvantage is lower protein yield, and it is expensive. This is the slide I was telling you that is common. So this slide is actually similar to a slide presented by Mauro yesterday. But the purpose of this slide here is to try to convince you that there is an increased demand on optimizing mammalian cell expression systems. Mammalian expression systems, mammalian cell lines like CHO and Azure, PHK, and HEC293 have become the dominant systems because of the nature of the recombinant proteins produced. And there is a demand, actually, the demand of biopharmaceutical outstrip supply, necessitating a significant increase in manufacturing capacity through the optimization of cellular productivity and product quality in a most cost-effective way. So this figure here just illustrates the pipeline from R&D to large-scale production. And each step of these steps is actually investigated and trying to improve the processes by different labs across the world. We are very interested in the first step here, design efficient expression systems. We are trying to use temperature-responsive inducible promoters. We are also interested in host cell engineering by modulating the messenger RNA stability. We are also interested in bioreactor and cell culture optimization by using subphysiological temperature cultivation to improve the quality and productivity of recombinant proteins. So we are asking the question, can an inducible promoter, culture protein, construct system be utilized to improve the performance of mammalian cells in culture? And we are motivated actually by the work done on the bacterial culture protein, which has been translated into bioprocessing. These vectors are so-called B-called vectors generated between collaboration between university, University of New Jersey, I think, and Takara. This is a 4 different version of the B cold. All of these vectors using the cold shock protein A promoter and 3' UTR and 5' UTR, and it's very efficient vector. But again, you cannot use this vector if you want to express your protein in mammalian cells. So the problem with mammalian cells is that we don't know a lot about it, fortunately, and this is because perhaps mammalian cells normally live in homeostasis and buffered against sudden change in environment and temperature. And most of the knowledge we have today is emerged from studies on adaptive thermogenesis called tolerance and the use of decreased temperature culture mammalian cells in bioprocessing and the production of recombinant proteins, and for medical clinics investigating the effect of cooling on organ transplant. And today, only a few proteins have been identified to be cold shock proteins. This protein include cold-inducible RNA binding protein and RNA binding protein motif 3. So what we have done is we have intensively investigated the transcriptional and post-transcriptional regulations of the cold-inducible RNA protein. And I will not show you all the deletion analysis and RNA stability studies, but what we found, we identified 2 promoters, the core and distal promoters. The core promoter is active at 37, and it's a cold shock responsive at 32 degrees. It produces a short 5' UTR, about 82 nucleotides, messenger RNA, and it's if you co-transfect it with CERP, you find stimulation of the gene expression. We have identified the distal promoter. It produces a long 5' UTR containing messenger RNA 125 nucleotides. It's irresponsive to exogenous CERP and independently exhibit close to high activity in transfection assays. We have also investigated the post-transcriptional regulations. And at 37, you get a significantly truncated messenger RNA, and we still don't know what's the function of this truncated transcript. After exposure to 32 degrees for 24 hours, you get a transcript with 82 nucleotide messenger, which is transiently stable up to 6 hours and start to degrade. At 32 degrees, we expose cells to 30 degrees for 6 hours. You get a substantially longer 5' UTR transcript 125, which is more stable at 32 degrees, and it's also efficiently translated at 32 degrees. And deletion analysis of the 5' UTR shows that it's exhibit internal ribosomal entry sites. So based on this, these 2 findings, we are now designing 2 different mammalian expression systems, and we are testing them to see if we can achieve an increase in productivity of recombinant proteins in mammalian cells. Another initiative we have now, this is in conjunction with the new biotechnology building, is to establish a transgenic animal biofarming center at CACST. Because of time's sake, I will not go into details because it's just an initiative in its infancy, but I thought ICGEB could contribute with their expertise to help us to develop such facility. So I will just skip this. Another interesting projects that we have, I just want to mention, is we have a very interesting project with the University of Cambridge. And the goal for this project is to develop a protocol to preserve mammalian cells in a lyophilized dry state, to increase the shelf life of mammalian cell storage. And this is particularly very important because every year we have a pilgrimage season, and there is a huge substantial mass gathering, and the Ministry of Health is the biobank, is substantial biobanking prior to that. And the idea, if we can establish a protocol to lyophilize blood and constitute for a longer period of time, and then just reconstitute prior to use, that will be very beneficial. Another collaboration we have with the Institute of Membrane Technology. I found this is interesting because it's another Italian institution. I know ICGEB is international, it's not Italian, but at least it's located in Italy. And we have successfully collaborated with them on membrane system in regenerative medicine and tissue engineering. And this is just a list of projects that we executed with them. Finally, I want again to thank ICGEB for organizing this outstanding workshop. I really look forward for a successful and fruitful collaboration with researchers from ICGEB. I want to thank each and every one of those guys who contributed to this work in previous slides. And finally, I want to thank His Highness Dr. Turki Al Saud, the President of CAST. He's a very enthusiastic guy, and he supported us before when he was a vice president, so he's continued to support us right now. Sorry if I took longer than I expected. So thank you very much again for your time.